Hello everyone. Let us continue with the chapter Sexual Reproduction and Flowering Plants Part 2. Now we are going to study about the internal structure of the anther. So when you just look at the transverse section of the anther, the anther is a four-sided tetragonal structure. In the previous video, we have learned about the structure of the stamen and the filament. Right? That is nothing but the anther. So, when you just look at the transverse section of the anther, it consists of a four-sided tetragonal structure. So, what is present in the four-sided tetragonal structure? The microsporangia are present at each corners in each lobe. So, this microsporangia, further it undergoes development and become the pollen sac. So these extend longitudinally when the pollen grains are developed inside the pollen sac. So when you just look at this transverse section, this anther, it consists of four walls. It is covered by, it is protected by. So here, so this is the transverse section of the anther. So here you have four microsporangia. So this microsporangia, when it undergoes development, it becomes a pollen sac. So here, when you just look at this anther wall, so this is the region, right? So this anther wall, it consists of three layers which helps in protection and dehiscence of anther. Whereas the innermost layer you call it as the tapitum, that helps in nourishing the developing pollen grain. Okay, so this is the outermost layer is called as epidermis, right? So this epidermis, it is always single. It is a single layer of cells where the cells are flat. Okay, and next layer is the inner second layer. So this is the second layer, okay, which is called as endothecium. Endothecium. So this endothecium, it is also single layered cells, okay, and then the cells have callose thickenings. The cells have callose thickenings. And one more layer, the third layer, is called as the middle layer. So this is the middle layer. So now what is the function of all three layers? These all three layers, it helps in protection. It helps in protection, and then it also helps in dehiscence of anther. What do you mean by the term dehiscence? Split open. Dehiscence meaning split open. So where? So here when you just look at this section, you have this connective tissue. Okay, so this is the connective tissue. I shall mark it here. Okay, so here this portion normally, or otherwise if you just look at the normal diagram, you have like this. So here only dehiscence, split opens. So these three layers, epidermis, endothecium and middle layer, it helps in protection and also it helps in dehiscence of anther. When the anther split opens, the pollen grains will be released. Right? Okay, now here there is another layer, the innermost layer. Okay, this is called as the tapitum. So I'll mark it here. This is called as tapitum. So now what is the function of this tapitum? It nourishes the developing pollen grain. It nourishes the developing pollen grain. This is very, very important point to be noted. It nourishes the developing pollen grain. Okay, and one more thing, what is the importance of this tapitum? It has dense cytoplasm. So here this tapetum, it has dense cytoplasm. I'll just mention here, it has dense cytoplasm and more than one nuclei. More than one nuclei. Now can you tell me when the tapetal cells will could become binucleate? When? During division, during the nuclear division, when it is not followed by cytokinesis, then the tapetal cells could become binucleate. Right? Okay. Now here these are the 4 microsporangia. So this microsporangia, later on it develops into pollen sac. So what happens here, inside this microsporangia, you have cells, right? So all the cells are same, all the cells are same. You call that to be homogeneous in nature. Homogeneous. What do you mean by the term homogeneous? Homo means same, all the cells are same. So this group of cells, what is present here, is called as sporogenous tissue. Sporogenous tissue. Right? So each cell has a tendency to undergo meiotic division. So when each cell of this sporogenous tissue, when it undergoes meiotic division, it forms microspore tetrad. What do you mean by the term tetrad? Tetrad means 4. Okay, so here microsporogenesis take place. Microsporogenesis. So what is meant by microsporogenesis? Microspores are formed. Okay, so each cell is called as a pollen mother cell. It is normally mentioned as a PMC, pollen mother cell. So when this pollen mother cell undergoes meiotic division, it forms 4 microspores. That is why microspore tetrad. These microspores, they will be together. Okay, when later on, when it undergoes maturity, what happens here? They get dissociate from each other, and when dehiscence of anther take place, these pollen grains will be released. So each microsporangia consists of several thousands of cells where it can produce many pollen grains at each dehiscence. Okay, right? So here, this is very important. So what is microsporogenesis is? The process of formation of microspores, that is nothing but the pollen grain. When does it take place? When a pollen mother cell, when it undergoes meiotic division, it forms 4 microspores or microspore tetrad. So today's class we have studied about the internal structure of the anther and about the microsporangia. Okay, later on this microsporangia, it develops into the pollen sac. So what happens here? So what are the 3 layers which helps in protection and the dehiscence of anther, and the inner layer is the tapetum which helps in nourishing the developing pollen grain. Okay. So if you like this video, share, comment and subscribe my YouTube channel and follow me on Facebook and Instagram. Thank you.